Hello and welcome to the webinar. I'm Chris Cantle, Council Member of the British Society of Soil Science and Chair of the Northern Soils Network. I'm delighted to welcome you all to our 6th webinar of the year on World Environment Day. Before I welcome our presenters, I would like to introduce the British Society of Soil Science as hosts of today's webinar. We're an established international membership organisation and charity committed to the study of soil in its widest aspects. We bring together those working within academia and have a growing membership amongst practitioners implementing soil science in industry and those with a keen interest in soils. We plan to host 10 webinars during 2024, so keep an eye out on our website for further details. Before we begin, just some basic housekeeping. As there are so many of you here today, all your microphones have been muted. We will be taking questions at the end of both presentations, and we will monitor these throughout the webinar. Please could you submit any questions you have by 12:50 p.m. to allow us to get through as many as we can. Although there is a raise your hand button, we won't be using this unless the presenter specifically asks for a show of hands. Today's presentation has also been awarded BASIS and NROSO CPD points. If you are registered with either body, please contact us directly after the event. Finally, please be aware that we are recording today's presentation. This webinar has been organised in partnership with Rothamsted Research, who have recently been confirmed as a new corporate member at the Society. We're pleased to be joined by James Clark, Head of Communication and Engagement at Rothamsted, who is going to provide an introduction to the organisation. Over to you, James.
Thanks, Chris, and good afternoon, everyone. First slide, there we go. We are Rothamsted Research. We're the oldest agricultural research institution in the country. I'm actually the head of communications, so I'm not going to talk in any detail about our soils work, just give you a very broad overview of who we are. Next slide, please. So we've been around since 1843, and we were basically set up by a Victorian polymath called John Bennett Laws. He basically grew up in the manor house here on Rothamsted Estate, became very interested in how plants grew and what created healthy crops. And started doing experiments in his bedroom. He dragged a bath up to his bedroom, started messing around with turnips and cabbages, much to his mother's dismay, and experimenting with dissolving bones in acid, and created the world's first superphosphate fertilizer, Law's manures. But nobody really knew at the time what nutrients plants needed in order to grow healthily. So he got into a partnership with a chemist called Henry Gilbert, and they set up the first experiments here at Rothamsted. And the idea was to just divide up a field and apply different combinations of NPK to the soil. They had a control strip, so it was very far thinking, and they had an organic manure strip, so they could compare the application of chemicals to what was normally used at the time, which was farmyard manure. That really was the basis of how modern agriculture started, really thinking in very, very scientific terms about how we could really maximize the yields from our crops. Next slide, please. So now we're basically largely funded by UKRI, the government agency, through the BBSRC Research Council. It's about £30 million a year, a few other sources of income coming in from various other agencies and foundations. We have 3 campuses, 2 main campuses, one at Harpenden, which is where I am at the moment, and the other at Northwick down in Devon, which is really our livestock farm. Harpenden is much more arable, and Andy, who you'll hear from later, is based down at the Northwick site. A couple of other small sites, Broomsmine and stuff like in Woburn, give us some slightly different soil types for us to work on. We're quite productive. There's more or less a paper with a Rothamsted author coming out more or less every single day of the year. So that's 300 a year, and around about 500 people from about 35 to 40 countries. Next slide, please. We do 3 things for the nation that nobody else does. The long-term experiments I've mentioned, these are incredibly influential in terms of looking at long-term environmental change, both for soils and for crops. Samples were taken from those experiments every single year. We've now got 1,300,000 samples going right the way back, almost 2 centuries. That gives us an incredibly valuable resource if you want to go back and look at how soil changes over time. We also do the National Insect Survey. We've seen a lot of coverage about decline in insect populations and whether that's a thing or not. We're probably the only institution in the country that has a very long data set going back to 1964, looking at what insect species are in our traps We have traps all over the country, and we're monitoring that insect biodiversity. So that, again, has been very influential. And the farm platform down in Devon, which is where we look at livestock, which is really answering the question whether grassland systems and livestock can be actually farmed sustainably. And it's Britain's most measured farm, we like to call it. Everything that's going in and out of the farm is being measured, including the greenhouse gas emissions, and obviously a lot of work being done on the soils. Next slide, please. And just to say that, you know, this is delivering vast amounts of data. So one of the reasons why we've invested a lot of time and effort in building up our capacity in things like machine learning and AI is we have data going back a long time. In fact, the very basis of scientific statistical analysis and experimental design, a lot of that work was originally done at Rothamsted here during the 20s and 30s. And that's given us really an ability to look at how things have changed over a long period of time. Good example is the broad bulk experiment, which is our longest running experiment. You see quite clearly there how things changed with the introduction of new varieties and the introduction of fertilizers, and how that completely altered our pictures of farming. We're now seeing other effects, which I'm sure Andy will talk about a bit when he does his talk, but an incredibly valuable resource that obviously we hope that many scientists will be able to use. Next slide, please. We do 5 big research programs for the government. These are really ways of kind of targeting the science to deliver impact. We're very much committed to making sure that our science doesn't just stay in journals and is actually pushed out and has an influence in the real world. So we organize our science around these 5 particular programs, all of which really contain elements of looking at soils within them. And the sustainability of soils. Obviously, we look at things like microbiome, we're looking at semiochemicals in soils, how are plants, roots, and microorganisms signaling to one another, and we look at the way that soils develop and change over time with the long-term experiments. And that's really a summary of what we do, and Andy in his talk will give you a lot more detail about the science. Thank you.
Thanks very much, James. That was a great overview. I'd now like to introduce our first presenter, Don Lobb. Don Lobb is a former farmer and long-time soil advocate from Canada. Don owned a successful farming operation in Ontario, Canada that became a model for sustainable management of soil, water and the natural environment. It attracted visitors from 6 continents and was a site of more than 50 major research projects, many of which focused on soil and water management. He has served as a consultant and on advisory boards for federal and provincial soil and water initiatives and for the University of Guelph. He also participated in development and delivery of the Environmental Farm Plan and has been an author for best management practices manuals and many soil and water management columns. Don has developed, applied and promoted soil care practices and the soil care ethic. His dedication, example and leadership have been recognised provincially, nationally and internationally, including the Canadian Conservation Hall of Fame. Over to you, Don.
The British Society of Soil Science is highly respected and widely appreciated, and so I thank you for the invitation to contribute to your webinar on sustainable farming and agriculture in relation to soils. The definitions of sustainable agriculture are as varied as the motives of the authors, and they seldom consider soil in a meaningful way. Yet, healthy, productive soil is where reliable and sustainable agriculture begins, and that underpins social, economic, and political stability. Therefore, my presentation will focus on sustainable soil management. I do so from the perspective of a career farmer who believes that we each have a responsibility to protect and constantly improve the soil entrusted to us. I worked hard at this and enjoyed real benefit and great satisfaction in the outcome. I will now share some of my soil experience to provide context for my observations on the choices we must make to improve and sustain soil productivity. By the end of the 1970s, I was facing serious soil degradation with compaction on clay soils and loss of organic rich topsoil on hilltops. Researchers confirmed my suspicion that tillage was the dominant contributor to my soil erosion. In fact, on these fields, tillage erosion was measured and by itself was moving in excess of 10 times more soil off hilltops than the universal soil loss equation would predict. I loved this farm and I wanted to make it a place that my family could enjoy and be proud of. So, with consideration for agroecology, I retired the most degraded land to trees. Windbreaks were planted for crop protection. Riparian areas were treed for stream protection and habitat. Surface water and root zone moisture management systems were installed to reduce surface water erosion and to increase crop yields. The most eroded hills were reshaped and covered with topsoil from depositional areas. Today, this would be referred to as landscape restoration, and it brought prompt return on investment. Cover crops provided soil protection and much needed organic matter. To address the devastating impacts of full surface tillage, I needed to completely rethink crop production. After considering and trying most other options, I chose to investigate no-till, a practice where the soil is left undisturbed except for a narrow strip where the seed is planted. The adoption of no-till was not my primary objective. It was a means to an end where 80 to 90% of the soil remains in a healthy natural state without biological or carbon disruption. No-till could also reduce erosion, compaction, and organic matter loss. Of particular value to my investigation were two field scale sites where I compared the conventional plow plus cultivation system with no-till. On those sites, to ensure a useful comparison, I always committed equal effort to maximize productivity on both the conventional and no-till treatments. These sites covered the range of field conditions found on my farm, and the work continued for 17 years. Here, I could measure my progress in no-till adoption while researchers monitored soil change and water management impacts. Immediately, I found that no-till planting impacted and must be complemented by change to all manageable crop production components. Often within the system, there would be a chain reaction of effects. Over several years, I tested the management of each component for both no-till and conventional tillage, and did so on both easy to manage sandy loam and difficult to manage silty clay soil. Over time, the greatest no-till benefits came on those clay soils. By focusing on adaptive soil management rather than manipulative soil use, I was able to improve soil health, soil productivity, and profitability. The quest for a reliable soil health indicator became the focus of much research activity, particularly on my tillage comparison sites. Earthworm midden density was an obvious indicator of soil health improvement. By year 12, researchers found that worm populations on no-till ground were more than 10 times greater than those on the tilled side of my comparison. Water-stable soil aggregates were the strongest indicator of soil health. For the benefit of a group tour of my farm, I took equal-sized soil samples from each side of my long-term comparison. I wanted to point out how much darker colored the no-till topsoil had become as our soil organic carbon increased. 2 months later, I returned to remove the signs and hardboard. The fall rains had washed away almost all of the tilled sample, while the no-till sample remained intact. This was a real-life slate test and an unintended demonstration of soil aggregate stability. In year 14, on June 2nd, we received 5¼ inches of rain in 6 hours, with 4½ inches of that in just 30 minutes. The whole landscape was under rushing water. The steel road culvert carried muddy surface water from tilled farmland. The plastic pipe is delivering surface water from about 25 acres of my no-till land. My water was clean, even though the soil was erosion prone and had a 50-foot elevation drop to the point where water entered the plastic pipe. This clearly demonstrated the value of water-stable aggregates. Edge of field water quality provides a dramatic measure of soil management commitment and competence. This matters to the downstream population, and it has real impact on the environment. Clean, edge of field runoff water should be the ultimate status symbol among serious farmers. Through these years, I learned much about the importance of soil carbon, the function and frailties of soil biota, and the full value of those aggregates. I learned that because carbon is essential to development of aggregates, a carbon gap is not tolerable. The sustainability of our food system is limited by carbon gaps that exist as a result of soil degradation and because most human waste, food production waste, and too much animal waste is not returned to the land where food or feed is grown. We must close this carbon gap by redirecting those wastes back to its soil of origin or through further carbon capture based on careful crop choices and cover crops. I learned that the biocommunity is enormous. Even soil health and human health have important biolinks. I will touch on 3 biota that are particularly critical to soil carbon and soil health. Earthworms collect plant residue, process it, and deposit high carbon castings down to 1 meter deep. Standard tests for organic matter and fertility miss much of this. Tillage devastates worm populations. Mycorrhizal fungi act as hair-like plant root extensions and bring soil-borne nutrient to plants in exchange for sugar and starches taken from those plants. They then exude high carbon glues that bind soil materials together to form soil aggregates. This process exists in nature. However, tillage destroys these fungi. Bacteria are essential to soil health and contribute to nutrient availability. Some bacteria are hyperactivated by oxygen introduced into the soil by tillage. This results in increased respiration of carbon dioxide to the atmosphere as organic matter is rapidly consumed, and that results in soil carbon decline. Once again, the problem is tillage, and the process is counter to nature. Nature maintains a bacteria-fungi ratio that supports plant growth, soil health, and soil stability. Any full-surface tillage, including very shallow or infrequent tillage, upsets that ratio. A BIOS reset often takes 5 to 15 years. On my farm, researchers found that this took 9 years or more. Thus, full-surface tillage is the Achilles heel of both mainstream and organic agriculture. We must do better, and we can do better. I learned that we can mimic nature to give soil resilience and ensure reliable and sustainable productivity. Native soil is highly aggregated and further stabilized with undisturbed root systems and plant residue. Over time, as water-stable soil aggregates establish, they provide an aerobic root zone to support bioactivity for nutrient cycling and carbon sequestration. They allow water infiltration, storage, capillary water movement, and soil drainability. And they provide soil strength and stability to resist slaking, crusting, erosion, and compaction. And beyond those benefits, we can expect reduced emissions of nitrous oxide and methane as soil aeration improves. For me, farm benefits included earlier planting opportunities and with that, higher crop yield. All factors associated with soil health must be in place for water-stable aggregates to develop. Thus, the degree to which soil is aggregated is our most certain measure of soil health. Aggregation can be measured using relative water infiltration rates or with an app called SLAKES that is now available through the US-based Soil Health Institute. My experience and observations are remarkably consistent with what we can learn from history. Population is always determined by food availability, and that is determined by soil productivity. People always settled on the most productive soil. As population exceeded the soil's natural carrying capacity, degradation began. Degradation was then compensated for, and food supply further increased with selection of more productive plants, with irrigation and drainage, with some use of manure, cover crops, and compost, but particularly through the use of tillage that aerates the soil and thus accelerates microbial activity for nutrient release from organic matter. However, tillage is a double-edged sword. It destroyed soil aggregates, and that brought erosion. As earthworms and mycorrhizal fungi were destroyed, the capacity for aggregate regeneration was lost. As organic matter was lost, so were water holding capacity and nutrients. Soil productivity was simply used up. This brought collapse of food supply, it brought political instability, and it brought population decline. All thriving civilizations claimed to value soil highly. However, through the ages, this sad story was repeated over and over and over again. This was the story of Mesopotamia, Greece, Rome, and many, many others. No civilization has survived the consequence of exploitive agriculture. New frontiers with new soil was their means to survival. Today, the UN FAO reports a sobering reality. We have now passed peak soil, the point where productive soil can no longer be replaced as quickly as it is being used up. We are now exploiting the last frontier. With current trends, when we reach peak population, we will have gained another 2.4 billion people while experiencing a net loss of 30% of today's productive soil. So where are we on today's population curve? The answer lies in how we manage our soil. Our choices will matter. Soil restoration can change the path of history. Important progress has unfolded on several fronts, with sharp focus on soil carbon maintenance and soil biota husbandry. The crop science community is quickly adjusting to biosensitive support for plant production and protection. For the first time in history, we have the tools and the technology to progress from plant-focused to soil-focused agriculture. We now know how to increase organic carbon, support biological activity, restore soil aggregates, decrease compaction, and improve water management. We can now increase soil productivity to reverse the soil degradation curve, and thus we have the opportunity to sustainably support a high population. Beyond my experience, our most progressive farmers have abandoned full surface tillage as a means to maximize and sustain soil productivity. For example, in 2010, after 18 years of no-till, Dean Glennie of Ontario, Canada, was corn yield champion with over 300 bushels per acre. He only used enough fertilizer for 150 bushels. Scientists investigated to understand how this could be. They found a dramatic soil and plant microbial shift, and at harvest time, there had been 100% use of root ball nitrogen in no-till versus only 70% in the tilled check. Plant available phosphorus during the growing season on the no-till was double that found on the tilled check. In Iowa, USA, Clay Mitchell makes soil care use soil care his first priority. Though recognized as probably the most technically advanced farmer in the world, he long ago eliminated tillage for no-till and cover crops, and he has strategically applied landscape restoration. In doing so, he reduced inputs, including fertilizer, by 30%, while increasing grain yield 3 times faster than the national average. David Hula in Virginia, USA, on land that has been farmed since 1607, replaced tillage with no-till in 1987. He then set world corn yield records 5 times and has reached 623 bushels per acre in 2023. Beyond reduced tillage, we can increase crop productivity by moving depositional soil back to eroded upper slope positions and thus reduce field scale soil variability. This is landscape restoration. It is our only quick means to restore soil productivity to eroded landscapes, and it has relative permanence if full surface tillage is abandoned. This should be our first option in addressing variable soil productivity. Serious work is now proceeding with the Soil Health Institute and McCain's Canada towards sustainable use in tuber and vegetable production. Clearly, soil restoration and sustained productivity are both possible and profitable. In conclusion, soil restoration and protection are now a matter of choice. Do we have the vision and the commitment to act on our responsibility to protect and improve the soil entrusted to us, the soil that underpins a truly sustainable agriculture? The stakes are high and the choice is ours. If we do not act, how would I explain that to my great-grandchildren? After all, we are using their soil and their food source. How would you explain no action to your offspring? I thank you.
Many thanks, Don. Just a reminder that we'll be taking questions from both speakers at the end of the session, so feel free to add any questions you have in the questions tab. Our next speaker is Professor Andy Neal. Professor Andy Neal is a research scientist at Rothamsted Research. His research is focused on understanding the integrated behavior of plant microbe soil systems, developing a theory of soil as an extended composite phenotype in the process. Combining modern techniques such as shotgun metagenomics and X-ray computed tomography, and borrowing heavily from the extended phenotype concept developed by Dawkins, the emerging processual view of biology and critical systems theory, he is working with colleagues to develop a radically different way of thinking about soil. Over to you, Andy.
Thank you very much. I'll just show my screen. So, and thank you also to Don. I was really interested to hear what you had to say, and slightly humbled in the sense that I do enjoy working with farmers, and I'm always very excited that we're doing something new, but you've just taught me that actually we're not doing anything new at all. You were doing it back in the 1980s. So that really, I did enjoy your talk a lot, and you've got a lot of experience to teach us all, I think. So thank you very much for that introduction. What I want to talk about really follows on very nicely from what Don's been talking about. Don was talking about the carbon gap in his soils, and that's something, it's not a term I've used before, but I think I'm going to start using it because that's really what I am very interested in understanding, and particularly understanding how microbial communities and soil as a system respond to organic matter inputs and the consequences for carbon gaps in soil. I work, James mentioned right at the beginning that Rothamsted has 5 strategic programs. I work in one of those, which is called Growing Health. And as part of that program, we have made a purposeful decision to work with farmers who are trialing different approaches to work in this sort of regenerative agriculture space. And in those discussions I've had with farmers that we're working with, of course, one of the biggest issues, as Don pointed out, was the influence of ploughing, the damage that tillage does. And the big question that a lot of people are wrestling with now is, how do we get by without using the plough? And a lot of people at the moment in the UK are relying on glyphosate to do that. But of course, they're very concerned about the consequences for that, the long-term consequences particularly. And so this is why the work I'm going to talk about to you today really came about, and why I think this is an important topic that we need to discuss as scientists, and together with farmers sort of come up with optimal ways of using min-till systems in the most environmentally positive ways that we can. And of course, what's at the center of this is really a trade-off. It's about how much damage do we do to soil by ploughing, and how much damage do we do to soil by eliminating plough usage, but looking at spraying glyphosate and herbicides. And of course, when society talks about glyphosate, it very rarely talks about glyphosate within that context. It talks about glyphosate usage on its own and the negative associations that has, but it never balances those risks with the risks that we know exist for tillage. And so I'm very interested in trying to understand those trade-offs and understanding how we should use the plough and or glyphosate in the best possible way. Now, as part of that, we've been developing as was said in the introduction, a different way of thinking about soil. I'm a soil microbiologist, but I'm particularly interested in how microbes live in soil and the sort of environment that soil provides for microbes, how that can be manipulated for good or ill by management. Now, that means we need to think about soil pore space. Soil pore space is where microbes live, particularly pores that are of the sort of size of 100 microns or less, so considerably less than the width of a human hair. But these pores are very important, not only as the place where microbes live, but they're also where root exudates tend to be delivered, where fungal hyphae tend to be most active, and where, if you choose to measure it, you find the highest levels of enzyme activity in soil. So this pore space is really where the rubber hits the road in soil. And it also, because of capillary action, accounts for a large part of water holding capacity in soil. So understanding the dynamics of these pore spaces, 100 microns or less, in respect to organic matter inputs and the consequences of these changes upon microbial community metabolism, at least the metabolic function that we can determine from metagenomics, is really at the heart of this work. And I want to start by just showing you some evidence of different soil behavior determined by texture that we've developed from the long-term experiments at Rothamsted that James mentioned. On the left is the total porosity plotted against the proportion of porosity that's connected. This data is collected in soil aggregates of about 2 millimeters because we're looking at such small pore spaces. So the connected pores are those which allow connectivity from the bottom of a 2 millimeter sized aggregate to the top of it, so allowing a free path through the aggregate. Now, what I want you to take from this is that under 3 different types of management, and here we've got a permanent pasture, so effectively a very lightly managed soil, together with 2 soils that are tilled, one of which is managed organically with NPK additions, and the other which is managed organically with farmyard manure additions, is that the total porosity and the degree to connectivity between those pores doesn't really change an awful lot. And in fact, the soil organic carbon doesn't particularly change an awful lot in these soils, and it ranges somewhere between 30 to 40 tons per hectare in the top 30 centimeters. In effect, these sandy soils on the left-hand side of this texture triangle do not adapt to the way they're managed physically. The physical environment in soil doesn't change depending on soil carbon stocks, depending on how the soils are managed by the farmer. On the right, and this is very consistent with what Don was just saying about where he saw the greatest influence of his management. Sorry, on the right, where we have soils which have a greater proportion of clay and silt, we see that soil actually is very responsive to the amount of organic matter in these soils. Now, let me just bring up my laser pointer. So this is a whole collection of soils from the Harpenden site at Rothamsted. So these are part of the long-term experiments that James showed you. And what I want to make clear is that soils in the bottom left-hand corner of this figure have extremely low organic matter because they've been treated as a bare fallow soil for over 50 years when this data was generated. So extremely low organic matter inputs, carbon stocks of about 20 tons per hectare or less. You'll notice that those soils have very little porosity and that there was also very little connectivity between them. As we increase our soil organic carbon inputs, it's not apparent from here what the carbon stocks are, but you'll just have to take my word for it that up as we approach to the very top of this curve, where we have the permanent pastures and the organic plots upon Broadbalk, we now have accumulated up to about 80 tons per hectare. And here we see a large amount of total porosity and very high connectivity between them. Now, what's particularly interesting is that this relationship is not a linear one. It's a non-linear relationship, and it has all the hallmarks of a self-assembled system and showing points of critical points of soil porosity around which we can observe tipping points. Perhaps the most important one is that at somewhere between somewhere around 5% total porosity, the evidence suggests that all pores in soil become completely disconnected. And we will look at the consequences for that in the next slide. But essentially, there's a strong relationship with organic matter in soil. So from the point of view of Don's talk, in closing that carbon gap and increasing porosity, and more importantly, connectivity between those pores in soil. So there's this clear relationship between organic matter and the physical structure of soil. Now, what does that mean from a practical point of view? Well, here I've got some images of soil aggregates. Their maximum size dimension is about 2 millimeters. And on the left here, we have a graph of net carbon flux through these soils. Again, these are from Rothamsted's long-term experiments, and the degree to which these pore spaces show connectivity between the pores. Now, the bottom aggregate here is generated from organic matter poor soils that was treated as a bare fallow. The top from a permanent pasture, which has upward of 80 tons per hectare of organic matter. So very different organic matter content, and we can see the consequences of that if we start looking at the structure that was within these aggregates. So in a minute, you will see the pore space appear within these aggregates as yellow. There's a sort of yellow skeleton inside there. And what you'll observe is that the permanent pasture in the top here, full of pore space, and all those pores are very highly connected. And if we look at the bare fallow soil here with less than 20 tons per hectare in the top 30 centimeters organic carbon, much less pore space, much less connectivity between them. So visually, this is very compelling to demonstrate that the way we manage organic matter stocks in soils can create a very different habitat for soil microbiology and soil invertebrates to live in. And also, of course, that changes the way plant roots can access water. And all that has got to do with the connectivity of those pores. Now, if we think about trying to live in these environments, then the next two movies give you some idea of the consequences for this lack of porosity and lack of connectivity in poor soils, the challenges that it faces or it presents to organisms trying to live in these systems. In the pasture up here, when we dive inside, you'll notice we have a lot of pore space and a lot of connectivity, so that there are many different paths through this soil aggregate. So access to nutrients, oxygen, and generally the movement of all the good things that biology requires is very free and consistent in this high organic matter aggregate. In the bottom here, very little carbon in this soil. You'll notice that we still find pores, but each pore tends to end in a dead end. So actually moving products, moving oxygen, moving water, moving all those important bits and pieces that microbiology and other microbes and invertebrates need to live in soil becomes a much more challenging task in soil that has very low organic matter. And this has a real consequence for the way that microbes evolve, or the communities evolve, and their metabolic activity in these soils. So if we go back to that original texture triangle and now look at what we see when we use shotgun metagenomics, we see that the non-adaptive sandy loam soils, so those that the physical structure doesn't really change as a consequence of management or as a consequence of organic carbon stocks, we really don't see much change, and these are in the functional genes associated with the microbiome. Now, I'm not particularly interested in you looking at all the details here. This is just a very high level look at the numbers of genes that we determine change significantly as a consequence of management. So again, the sandy soils are not only non-adaptive from a physical point of view, but they also seem to be relatively non-adaptive from a biological point of view as well, as opposed to those soils on the right-hand side here, where we were, if you remember, able to demonstrate a very large change in the physical characteristics of soil associated with carbon. We also are able to see very large changes in the microbiological function determined by the genes in the microbiome as a consequence of these changes. Now, what's particularly interesting is that very few of those genes are actually responsive to the organic carbon stocks themselves in the soil. Many more are sensitive to the permeability, which is determined by the degree to which this pore space allows nutrients, oxygen, water to move through the system. So we have a much greater influence upon microbial activity by considering the pore spaces that we generate than by concentrating solely upon the organic content of the soil. And the amount of genes subject to sensitivity to this permeability are very similar to the number of genes that we observe to be sensitive to the management. And in this case, again, it's contrasting permanent pasture with soils that are fertilized either with farmyard manure or inorganic fertilizer. So the structure of the soil would appear to be, or the structure of the soil that you create would appear to be influencing as many genes as the very broad scale management of should we have an arable system or should we have a pasture system. So this permeability is a very strong link to the function of the microbial communities in soil. This led us to suggest the theory of soil as actually an extended composite phenotype of the microbial metagenome. And this effectively says that soil is a product of the genes within it, manifest through the combined effects of multiple organism phenotypes. And what we mean by that is this soil structure, as Don was explaining earlier, is a function of microbial activity upon the organic matter that is driven through the soil by farming management or by management in general. And that this organic matter directly relates to organic pore structure, which controls connectivity between pores, and as a consequence, the hydraulic conductivity, where pores soil with very low carbon, have very poor connected pore space and very low hydraulic conductivity, whereas as you increase organic carbon, you get a much more connected pore space and a much more connected and hydraulically conductivity active system. This then places constraints upon the microbial community in those soils, largely by virtue of its resistance to oxygen movement through these soils. We've shown that in that bare fallow soil, the very degraded soil as a consequence of carbon starvation for 50 years, that even at field capacity, with models using these aggregate structures, that about 30% of that pore space was anoxic, even at field capacity. and this reduces to only about 5% in the pasture. So the control of oxygen access seems to be one of the principal ways in which pore space and the permeability of oxygen through these systems influences microbial activity. So the point that Richard Dawkins made is that we're not just looking at the structure that is formed, but actually that this then, by various reasons, reflects back and has selective pressure upon the microbiome. What we have noticed is that in soils that are less connected and have less pore space, as a consequence of reduced oxygen availability, that genes associated with anaerobic respiration become more abundant in those communities. Because there's less oxygen, microbes have to use alternative electron acceptors. Of course, one of the big ones that we use in agriculture is nitrate. And so we see genes associated with denitrification and genes associated with the production of nitrous oxide as a potent greenhouse gas become more abundant in soils that have very low connectivity and very low carbon. So the connectivity is a consequence of having very low carbon. So this is what this carbon gap, as Don put it, does to microbial activity. It causes it to shift towards negative from the point of view of nutrient use efficiency and from the point of view of greenhouse gas production from soils towards these negative behaviors, because we are effectively, by limiting organic matter inputs to soils, we are limiting the way in which oxygen can move through those systems, and the microbiome evolves as a consequence. Now that's one explanation for why organic matter is so important for soil fertility. Another is, and then this can seem very abstract when we're talking about metagenomes and we're talking about extremely small pore space, but some nice work in the United States has shown that there is direct relationship between soil organic matter, its capacity to hold water as a consequence of this increased porosity, and the insurance payouts under drought that are recorded in the US Corn Belt. And essentially, there is this relationship between high soil organic matter and low costs of insurance payouts under drought. And if you look on the right here, these yield graphs show that the difference between low organic carbon in red and the high organic carbon in blue only really becomes apparent under severe or very severe drought. But once you get to this level of stress, the effects of high organic matter in soils creating that pore space of these scales, which is effectively creating that water holding capacity, you can start to see these effects under severe stress at continental scales. So although, yes, we're working very abstract and in scales that are very difficult to see by the human eye and dealing with the pore space in soil of that scale, we can see that this sort of behavior in soil has very important implications, not just for the way we grow our crops, but financially in the way that insurance payouts are at the behest of drought systems in the US. So I hope that has laid the ground for saying what we want to develop in soil, and you could say soil that is in its best state has a high organic matter content, and as a consequence has high porosity, high connectivity between those pores that allows nutrients and oxygen to be delivered rapidly to biological activity in those soils. So keeping that carbon gap, as Don referred to it, as small as possible is really important. And if you start thinking about how regenerative agriculture is being practiced now, a lot of it is directed directly at how to minimize that carbon gap. And here I just have some examples. Should I be seeing myself on the screen? No, it doesn't matter. Okay, so we have some examples of the consequences for different management upon organic carbon inputs and organic carbon stocks in our soils at the Northwick Farm Platform. Now, it was managed since 2012 as a pasture system. Sorry, I'm trying to get rid of my window so I can see what I'm talking about. I'm going to close it and hope I don't get rid of myself completely. Okay, so since 2012 up to about 2019, it was managed as two contrasting pasture systems, one which was unimproved and so had no tillage or reseeding, and a set of that was in blue, and a set of pastures that were improved and were ploughed every now and again to reseed clover in there to reduce the amount of nitrogen, bagged nitrogen, that was added to those soils. Now the top graph
Andy,
shows
sorry to interrupt, Andy. Just time check as well, just to give you a reminder.
All right. Then I will briefly run through these. The point I want to make is that in 2019, we switched some of those plots to an arable system, and we reduced the amount of photon energy captured by those fields from about 4 millijoules per square meter per day down to about 1 megajoules per square meter per day, as a consequence of not only the crop phenology, meaning that it was when most sunlight was available in the UK, the crop had already senesced, and also the fact that after harvest, the soil was left bare. So when it comes to carbon inputs, conventional ways of managing arable systems results in a real reduction in carbon inputs to the system. And in the bottom right-hand corner here, I just want to show that as a consequence of ploughing, and of course, we're all aware of this, as a consequence of ploughing large amounts of organic matter, even from one ploughing event. So this is permanent pasture, and this is the arable taken out of permanent pasture. After 1 year, it had lost nearly half of its organic carbon stock, or content, sorry. And that it took about 8 years of non-being disturbed to get that carbon level back up to the original soil organic carbon. So understanding how we transition, how we get organic matter into soil by keeping it covered and keeping green plants, so cover crops essentially, after the crop has been harvested, but do that without ploughing, and so don't disturb that organic matter stock, don't disrupt that texture and that pore space. That's pore space is what I meant, is really the trick. And so the reason why spraying is now becoming such a big issue. Why can I not move on? For some reason I can't. There we are. So we've been working with Ian Wilkinson at Farm Ed, which is run as a farm demonstration project. This is X-ray computed tomography of, on the left here, a 4-year herbal lay. And Ian runs Cotswold Seed, so he's very good at producing extremely species-rich herbal lays. And you can see that after 4 years, this herbal lay, so this is the pore space in here, is highly porous, lots of connectivity, lots of different sizes of pores. But after 4 years, after a ploughing event to take that herbal lay, or remove that herbal lay and go into a winter wheat, you can see the consequence of that ploughing has drastic effects upon the porosity of these soils. So all that good porosity and connectivity that is keeping oxygen available in these soils is largely lost as a consequence of 1 ploughing event and 2 power harrow passes. Compare this to what he observes in his min-tilled and glyphosate-treated cereal crop, which he keeps as a control, and you can see that there's much more porosity being maintained in that soil as a consequence of this management. And if we try and put metrics on that, we use Euler density, which is effectively a measure of connectivity, and counterintuitively, the more negative the number means the greater connectivity we see between pores. We can see that the consequence of the post-herbal lay winter wheat ploughing is that we've lost a lot of the connectivity of those largest pores in those soils. So having an alternative to ploughing to get out of herbal lays or cover crops is really important for maintaining soil structure, organic carbon stocks. Now, I think what I'm going to rush through these. We've done some work with the Wild Farmed Growers Collective, and also with Tim Parton, who's part of the Green Farming Collective, to look at the degradation rates of glyphosate. It's very preliminary, but we've seen some very important consequences. Now, the important thing to understand is that glyphosate is broken down aerobically and anaerobically, but the rate of anaerobic degradation is much slower than it is with lots of oxygen availability. So again, this argues for a soil having a high organic matter content, high porosity, high connectivity, and high permeability of oxygen through that system to degrade any glyphosate used as rapidly as possible. The top here, just to cover it very briefly, we did observe changes in his soil from a zero-till system to one that was cultivated to 10 centimeters. The first week saw an increase in pore size, but then a slump 7 weeks later, so that a lot of that pore space was lost as a result of tillage. And we saw that even in the zero-till systems, that the glyphosate, as best as we could manage it using LCMS, had been degraded after about 28 days. So the persistence of glyphosate in these soils is no more than about a month, but it seems to be accelerated in these till systems where you have more oxygen in the surface. It seems to be degraded more rapidly. I'm just going to very briefly go over the evidence. There's very little evidence that glyphosate has long-term effects upon microbial activity. This is the results of a meta-analysis published in 2016 that suggested that microbial biomass has some no real trends in response to the glyphosate concentrations added to soil, but soil respiration certainly does. And this is probably a reflection of the extra effort that the microbial community is having to go through to degrade glyphosate, and some measure of the stress of glyphosate addition. But if we look from US long-term experiments, and there's 3 here that are based upon soils in Eastern Washington, Beltsville, Maryland, and Stoneville, Mississippi, and I don't want to go through all the multi-dimensional analysis particularly, but the outcome from these 2 experiments performed in the US on long-term managed soils is that the effects of glyphosate are effectively difficult to determine when you look in real soils, and that issues such as the proximity to the rooting system, so the rhizosphere effect essentially, or fluctuations from one year to the next, or the general organic versus inorganic management have much greater influence upon the soil microbial communities, and this is really who's present and who's absent, not function, than the application of glyphosate. If there is an effect of glyphosate, it's much less than other processes that are going on the farm at the same time. So I will just finish up by essentially summary of everything I've said. This won't be new to anyone listening to this, I'm sure, but organic matter is essential for optimal soil function. And we see this texture-dependent critical behavior where clay soils shall have much more response, so much wider responsive range to organic matter inputs. That high organic matter in these responsive soils gives you high porosity, high flux, and high nutrient use efficiency, as well as water holding capacity. And regular tillage is associated with losses of these functions. So of course, the min-till approach has all these benefits of not causing degradation, of not seeing these shifts in microbial community respiration. Glyphosate has effects upon microbial communities, but they're typically less than what we see in the field for other management approaches. So we end up with this idea that if we're going to use glyphosate but in the most sensible way, that we should have soils that are already in a good, healthy state, maximizing microbial activity, microbial biomass, and most importantly, microbial aerobic respiration, because that's where we get the most effective degradation of any glyphosate additions. My one point of caution to end on here is that at the moment, we have no idea on how glyphosate additions over the long term change microbial community function. And that's where our research is going in. That's the direction we'll be taking in the next few months and years. So with that, I shall finish. I hope I haven't gone over by too much.
No, it's fine. Many thanks, Andy. That was really interesting. So yeah, thanks very much. As we are running slightly behind, hopefully see in the chat that we will carry on until 1:15 to answer as many questions as we can. So we now come to the Q&A section of the webinar, which will be led by BS Cubed Early Career Committee Member and Soil Scientist Consultant at Arcadis, Jay Ryan. Over to you, Jay.
Chris. So I'll start off with my first question directed to Dom. What is your observations of weeds in a no-till system?
Observations of which?
Of weeds?
You mean before I started to see some benefit?
Yes, like did you have much experience with like issues with weeds when you adopted your no-till?
Weeds. Okay. The weed pressure that we have with the tilled system has become tuned to the tilled system over time. They're the weeds that adapt best to a tilled system. When we switched to no-till, then very soon the weed pressure actually dropped because we were no longer planting those weed seeds with tillage. And to come back to what Andy was talking about with glyphosate, I used glyphosate as a burn down, not all the time, but most of the time. And in spite of that, we still had the huge improvements in soil aggregation that I showed you. And we had the increase in earthworm populations that I showed you. So the glyphosate certainly wasn't having a negative impact on those fronts.
And also, Dom, originally, there's a bit of a misconception that no-till can have a bit of a reduction in both productivity and profitability. It's clear that there are benefits that cannot be ignored. But when you first started to adopt this approach, did you experience some of these reductions?
When I repeated what the research community had been doing, I got a 30 to 40% yield reduction. When I moved beyond that, I was getting my highest yields with no-till. Now, when I started working with it, the research community here was saying that we could expect a 30% yield reduction. And so when I first started trying the practice, I took a look at what they were doing, and they were working with continuous corn or maize on clay soil or clay loam soil. But when I tried and where I repeated that, I got the same yield reduction they did. But where I planted no-till maize or corn following out alfalfa grass sod, I had the highest yield I had ever had on the farm. And that comes right back to what Andy was talking about. If we're going to start into a practice like that, if you start into soil that's already is highly aggregated and in good health, there's no need to put up with that yield reduction. But almost all of the research that have been done on no-till have been done on sites that were only within 3 to 5 years long. And you don't get that bioadjustment and aggregation development in that shorter time length. And it almost always been done without a proper crop rotation. And so almost all of the research, if you look at it, will show that yield reduction, but it's not necessary.
Over to you, Andy. Can you go wrong with applying any ample organic matter, in particular fresh manure? And can you still receive poor porosity as a result of this? And what is likely the causes if you are still seeing the poor porosities after application?
So I think that the fact that we see a strong influence of texture upon how a soil responds to organic matter input suggests that, yes, you could go wrong in the sense that you could apply organic matter to the soils that have the sort of texture that isn't going to respond to it. So I think understanding your texture, so effectively your sand, silt, and clay, which any analytical lab should be able to provide, tells you whether it's a good investment to put organic matter in that soil. So that's if it's got a good clay silt component, or whether it's a poor investment and most of it is going to be but respired away. So I think if you're putting organic matter into soils but not seeing your porosity change, you might stop and think about what texture it is first, and then reassess whether it's necessary. I mean, also, if you've got a farm that has many different soil textures, you might choose that there are certain textures that are well worth putting organic matter in, and that some parts of the farm probably will not benefit an awful lot from that investment. So yes, I think it is easy to go wrong, but once you know what your texture is, and then you understand something about how this drives organic matter sequestration, then I think you can correct that relatively easily.
Follow on from that, what is your opinion on the application of biochar, and how is that likely to affect the porosity as well?
So I have to say I haven't worked with biochar, so all of this is my own personal opinion. They've got no evidence to support this, but our, the way we're developing our theory of how we think soil works as a biological system is that it needs organic matter to drive it. And although biochar in the sense will be very good for your carbon measurements, it's mostly relatively inert organic matter inputs. And so from the point of view of driving microbial activity, driving this structure formation, which requires sort of necromass and biomass to stick soil together, it's probably less effective at creating this structure because it's not driving that microbial activity. So I guess my point of view is it's not driving a healthy state, it's just really a way to capture carbon. But that may be fine if that's what you want to do in your soils.
For Don, how do you deal with organic soil amendments such as farmyard manure, for which the carbon will be lost to the atmosphere if left on the surface without incorporation?
That will always be a problem. And I know that people who are dealing with liquid manure typically are injecting it because of problems with odor and that sort of thing. But that injection process is very, very disruptive to the soil. I guess that's a challenge where we're going to rely on maybe the Andes of the world to help us find ways to apply that manure in an efficient way. But on the other hand, that's the way nature dealt with manure. It was always applied naturally to the surface, and so maybe that's something we learn to live with and offset that effect as much as we can with other practices that we perform. That's probably not helpful, but that's one of the challenges we have. Soil management very much has to be addressed by the science and art of soil management. And as a farmer, I was one of the artists who had to pick through the scientific silos and find those things that would combine to provide the best results. And you've touched on one of the challenges there.
So as a farmer, how do you feel that soil scientists can best help communicate with farmers about the importance of soil health and how much they need to prioritize implementing some of these on their farm?
Well, in my case, I simply proceeded in the face of the scientific advice that farmers or the scientists were suggesting that no-till is going to produce a yield reduction. But then the scientific community came on board and learned as much as they could from me and from what I was doing. But the most help that the scientific community could provide would be to help us put together the crop production systems. And I showed it in the one slide I had. There was a circle with all of the manageable components of that crop production system, and every one impacts the others, and you'll get a chain reaction of effects. And if they can help us put that together, then we can move together quickly. And it was when I put that together and started advocating that we started to make some real progress here in this country, and I know some of our American friends picked up that concept too, and people like Dwayne Beck from South Dakota State University, who travels the world, picked up on that concept, and that's where we really need help, is to put that together.
Sure. Back over to you, Andy. Is it the case that a soil can be turned from non-adaptive to adaptive by increasing soil organic matter content and improving microbial activity?
I don't think so, because I think that the adaptive versus non-adaptive is driven by the texture. Now, what's very interesting is I was speaking to a farmer many years ago now about the problems associated with sandy soils, and he said, it's funny because my grandfather used to go, he just waved in the air with his cart and used to dig up clay and bring it back to these sandy soils and used to add it. And I thought he was completely balmy, but he swore that it did good things for his soil. And I think that's what was driving that farmer's behavior was, yes, if you've just got a sandy soil, you're not going to change it unless you do something about the texture. That person's response to was, well, I'm going to add clay, right? Now, that may be easy if you've got a readily available source of clay close by, but for most people, I think if you've got a sandy soil that's not responding to organic matter additions, you have to find other ways of managing your system. You're not going to be able to change that basic fact. The problem is, because say sand particles are so big, they leave a lot of space between them for oxygen. So there's always readily available, I know it's counterintuitive to what I was saying, but there's no way to lock organic matter up so that it becomes unavailable for microbial activity in a sandy soil, because there's so much, the pore spaces are huge. Whereas in a clay soil, you do get this occlusion and this sort of sealing up of organic matter away from microbial activity. That's the problem. Unless you can create that in a sandy soil by adding smaller particle sizes, I think you will always face the challenge, and it will never be an adaptive system.
Could I comment on that one too, please?
Yeah, I'd love you to.
Okay, I think what you're talking about, Andy, is very close to what I was talking about with landscape restoration. And I have a son who's a soil scientist who has done a whole lot of work with that. And it actually began on my farm, but we're taking topsoil from depositional areas and putting back up just a thin layer up over the eroded soils. And quite often those eroded soils on hilltops were a coarser textured soil.
Yeah.
And by doing that, they could bring that eroded soil productivity back to at least 90% of what the most productive parts of the fields were. And this has been going on for some time, and I've seen pictures back to the 1930s where people in France were doing that.
That's interesting, yeah. This is why it's so important to work with farmers. You have evidence, your practical evidence of some of the theoretical stuff we like to talk about.
Well, we need to work together always.
Absolutely, absolutely.
I've had the great privilege of working with some really good scientists.
Well, and they've had the privilege of working with you.
Thank you.
I've got another question for you, Andy. The genes responsible for permeability, are they highly localized to the soil, or would you expect the same genes to be expressed in a lot of healthy soils?
Okay, so I don't think there are genes specific for creating permeability, but the point I make there is that this permeability is driven by the organic molecules that are basically generated from microbial activity, which interact with clay particles because of the charge, and then they start acting as glue, rather like mortar in a wall. So there are no specific genes for sticking soil particles together, but when cells die, and those lipids, the term, if you look in the literature, you'll come across the term necromass. And it's that necromass, which is basically all those dead cells and the fragments of dead cells, which are responsible for starting to create this structure by virtue of sticking mineral particles together. Now, of course, where you've got more biological activity, you have more necromass. And so it's that structure that then builds as a consequence, and that feeds back on selection of genes. And I wouldn't say there is a particular gene for building structure. It's just driving that microbial activity, which sort of feeds back to why I don't think biochar is how you drive that. I think you drive that with fresh material that is labile and is driving metabolic activity. So yeah, it's not a genetic sense. It's about activity, driving activity.
And Don, what soil health indicators should scientists look for to better understand soil health and create an index for it?
Improvement in soil aggregation is number one. And that comes back to what Andy was talking about, where exudates from decaying biota of various sizes stick together the organic and mineral materials to form little crumbles, and you have micro aggregates, and I think Andy was referring to those mostly, and then we'll end up with exudates from mycorrhizal fungi in particular that stick together the microaggregates to form macroaggregates, and then we start to get real porosity in the soil, and when we have that, then we get good water infiltration and aeration that allow the microbes to function, and when that happens, then we have productive, healthy soil.
Now I got a couple for both of you. Now, what are your views on soil organic matter being over-reliance as a go-to measure for soil health?
Soil organic matter is different than stable soil carbon. The carbon is processed organic matter. And typically organic matter is somewhere in the order of 50 to 58% carbon. And it's only after that organic matter is processed that we get some soil stability. If you have loose organic matter, and this is where some of our organic friends get into problems, where if you have loose organic matter, that doesn't provide soil stability, you can still get soil erosion, and that's not a, it can provide some absorptive benefit for helping to hold moisture, but it doesn't allow the water infiltration that we need and give soil stability.
And I think you're right, Don. That separation in the two pools, I think, is really important, because we tend to measure stocks in soil, but actually we see this very strong relationship between organic inputs and the sort of structure that Don and I are both talking about, that is a good soil state. So yes, we're measuring stocks as a proxy, but actually the real relationship is between keeping organic matter flowing through the soil as long as possible. And, you know, because the further north you get, the less easy that is. But having cover crops, having intercrops, you know, having something growing in the field all the time. But yes, we have to measure something, and I think carbon is actually a pretty good proxy for the reasons that I've demonstrated, that we do see these relationships. But of course, again, texture is really the context that we need to think about this. Yeah. Does that answer the question? I think we did a good job, didn't we, Don?
Well, that has been my experience. So earthworm middens density on the soil surface gives a casual clue as to how healthy the soil is, because that's a pretty crude demonstration of bioactivity in the soil, but it's somewhat helpful. But Otherwise, it's if you can look at the edge of a field during a rainstorm event and find clean water running off of it, you know you have a healthy soil.
And I think we might have time for just one more. So I've got one more for both of you. What are your opinions that root vegetable growers to increase their soil organic carbon as they cultivate their soils?
Do you want to go first, Don? You're the farmer.
So you like to ask the really tough questions now.
I have it for last.
And the Soil Health Institute in the U.S. and McCain's Canada, who are very much into potato production and processing and that sort of thing, are really doing some detailed and intensive work on that right now to try and find ways to be able to maintain soil health during the production of those crops, because with the immense or really intensive soil disturbance that we have in the harvesting in particular, that's counterproductive to soil health. We may need to establish a different production paradigm altogether for how some of these crops are grown, and we may even change our diet some if we're really concerned about maintaining a food source.
I would just say because those root vegetables require such aggressive soil disruption, you would have to put in an awful lot of organic matter to try and fix that. The other approach, as done, I think you may have been alluded to slightly, is just grow them in sandy soils where disrupting them causes relatively less damage. But certainly in our farmyard manure plots on Broadborg, where we use stupid levels like 35 tons per hectare of farmyard manure, but it's ploughed every year, we have very high stocks equal to permanent pastures, and the aggregate scale pore structure is equivalent to grassland. So that suggests, were you to have sufficient organic matter to hand, which no farmer practically has, that you can disrupt and maintain a certain level of important structure. But it's beyond the ability of practical farming to do that. So I think you have to be very careful about where you do it and how you grow root vegetables.
Okay. Yeah, thanks all. Sorry, go on, Jay.
You're welcome.
I was just going to say thank you very much for answering the questions. I'll hand over back to Chris to tidy us up.
Yeah, thanks very much, and yeah, really interesting discussion. So thanks all, and thanks for sticking on a bit longer past 1 o'clock. So yeah, just to wrap up, on behalf of the British Society of Soil Science, I'd like to express our gratitude to Don Lobb and Neal for coming along to present today, and also to Jay Ryan, James Clark, and Rothamsted Research for supporting the webinar. Thank you all for attending. You will find a quick feedback survey when you leave the webinar, which we hope you will take the time to complete. The recording of this webinar will also be available after the event on our YouTube channel. Our next webinar is scheduled to be on Wednesday, 3rd of July on soils and water, which will be led by the BS Cubed Early Careers Committee. So keep an eye out on our website for how to register. I hope to see you at future events, and in the meantime, thank you again and goodbye.